Assalamu alaikum class. Welcome again to the series of lectures on the extension and modifications of Mendel's laws. So far, we have discussed the phenomenon of codominance, incomplete dominance, and multiple alleles with relevant examples. In continuation to our discussion, today we will learn about lethal alleles, penetrance and expressivity, and about gene interactions on which we will focus specifically on epistasis and its types. So let's begin. Learning objectives. Today I'll be describing the recessive and dominant lethal inheritance patterns. We will be understanding the phenomena of penetrance and expressivity, and I will also be explaining the phenotypic outcome of epistatic effects between genes. So first of all, we have lethal alleles. Lethal alleles are also referred to as lethal genes or lethals. These are the alleles that cause the death of an organism that carries them. They are usually a result of mutations in the genes that are essential for growth or development. Lethal alleles may be recessive, dominant, or conditional depending on the genes or the genes involved. Lethal alleles can also cause death of an organism prenatally or at any time after birth, though they commonly manifest early in the development. Now let's look at the inheritance pattern of the lethal alleles. A large proportion of the genes in an individual genome are essential for survival. You all know that. Sometimes a non-functional allele for an essential gene can arise by mutation and be transmitted in a population as long as an individual with this allele also have a wild-type functional copy. The wild-type allele functions at a capacity which is sufficient enough to sustain life, and it is therefore considered to be dominant over the non-functional allele. However, consider two heterozygous parents that have a genotype of wild type and a non-functional mutant allele for a hypothetical essential gene. Now, in 1/4 of their offspring, we can expect to observe individuals that are homozygous recessive for the non-functional allele. Because this gene is essential, these individuals might fail to develop after fertilization, and they may also die in utero, or they may also die later in their life, depending on what life stage requires that particular gene. Inheritance pattern in which an allele is only lethal in the homozygous form and in which the heterozygote may be normal or have some altered lethal non-lethal phenotype is called the recessive lethal allele. A very good example is of the Manx cat that you can see on the left side. Now, for the crosses between the heterozygous individual with the recessive lethal allele that causes death before the birth, when homozygous, only wild type homozygotes and heterozygotes will be observed, and they are the ones that are going to survive. And the genotypic ratio is therefore going to be 2:1. In other instances, the recessive lethal allele might also be exhibited as a dominant but not lethal phenotype in the heterozygote. As I told you, the example of curly allele in case of Drosophila, in which the heterozygotes are, they have that curly wings, but it is the lethal in the form of a homozygote. Another example of the recessive lethal allele occurs in the Manx cat, the cats which either don't have a tail or the tail size is really short. Now, the Manx cat possesses a heterozygous mutation which results in this shortened or missing tail. Crosses of the two heterozygous Manx cats result in 2/3 of the surviving offsprings that display the heterozygous shortened tail phenotype, and 1/3 of the surviving offsprings of the normal tail length that is homozygous for a normal allele. Now, homozygous offsprings for the mutant allele cannot survive birth, and therefore they are not seen in these crosses. Then we have the another pattern of the inheritance of the lethal allele, that is the dominant lethal alleles. Now, when a single copy of the wild type allele is not sufficient for normal functioning and even for survival, the dominant lethal allele inheritance pattern is the one in which an allele is lethal both in the homozygous as well as in the heterozygous condition. That means that the allele can only be transmitted if the lethality phenotype occurs after the reproductive age. Individuals with mutations that result in dominant lethal allele fail to survive even in the heterozygous forms. Dominant lethal alleles are very rare because, as you might expect, the lethal alleles only last one generation and they are not transmitted. However, just as the recessive lethal alleles might not immediately manifest the phenotype of the death, dominant lethal alleles also might not be expressed until adulthood. Once the individual reaches reproductive age, the allele may be unknowingly passed on, resulting in a delayed death in both generations. A very good example of that in case of human is the Huntington's disease, which is a disease of the nervous system in which the neurons gradually waste away. People who are heterozygous for the dominant Huntington's disease allele, which is capital H and small h, they are inevitably develop the fatal disease. They are going to develop that. However, the onset of the disease may not occur until the age of 40, at which point the affected individuals may have already passed the allele to almost 50% of their offsprings. Now, to understand the lethal alleles in detail and their inheritance pattern, let's watch a very nice video by Professor Matthew Smith explaining the experiment that actually led to the discovery of lethal alleles.
Hello, I'm Professor Matthew Schmidt and welcome to Genetics. We're continuing our series of revisiting Mendel, again in the sense of wondering when we see aberrant results, or at least those that seem odd, does this mean that there's a problem with Mendel's laws, or how are we going to interpret this so that they're consonant with his laws and explain the apparent discrepancy? So this session is a little shorter than some others. It really addresses the phenomenon of lethal alleles, and I wanted to put the idea of pleiotropy in there as well. So let's see what this is all about. It turns out that we're going to see the definition of a lethal allele, but I want to explain to you the experiment that led to the discovery of this. In mice, there are two alleles that exist at one of the loci that controls coat color. Now, coat color in mice is actually a little more complicated than we've let on, but this is the same locus that we were talking about in a previous video that had to do with being agouti versus black, etc. So an allele was discovered that was actually dominant to agouti, and they called it capital A, capital Y to give the idea that it was dominant. Now, it caused a yellow coat color, at least this is the preliminary story, and it turned out that if they took two yellow mice every time, no matter what, two yellow mice, crossed them together, what you see is a strange 2-to-1 ratio of yellow to non-yellow. It turns out that the non-yellow is agouti colored. But a 2-to-1 ratio, guys, it's certainly not 3-to-1. There's no way to make, which is why I put this separate, there's no way to think of a 2-to-1 ratio in the context of 9-to-3 to 3-to-1. It really just doesn't work. No way I can see. So what is the explanation? Well, it turns out that all yellow mice are heterozygous, meaning they have one capital A, capital Y, and one regular capital A. You would think under normal circumstances that a yellow mouse could be capital AY, but we're going to see that that's not possible. But first, let's look at the experiment. So the idea was, after doing some preliminary stuff, I mean, it's sort of a little backwards the way we're thinking about it, but the fact is all yellow mice are heterozygous, but how did we determine that? So if they took a pure breeding agouti line like this and crossed it with the yellow mice, always what they saw was that you got 50% yellow and 50% agouti. We know that this parent can only contribute the big A allele, right? So what that means is the other parent would have to be contributing the big AY half of the time, and big A the other half of the time. So every yellow mouse that was in existence, when they crossed it like this, they always got the 50-50 ratio, which implies strongly that the yellow mouse is in fact heterozygous for this AY mutation and the regular agouti allele. So what is going on here if that's the case? Well, if you do the cross yellow times yellow, so now we know what that means that, sorry, thought I had my highlighter. Each parent can donate a Y and A, right? So let's follow that. A Y and A Y and A. And let's just fill in the boxes. So we get one homozygous dominant. We get two that are heterozygotes for AY and A. And we get one that's capital A, capital A. So this one looks agouti. As you can tell, this one and this one look yellow. And what's up with the other one? Well, it turns out this little cross through it means that these type of organisms, while they can be conceived, they die before they even implant. I mean, it's you never will see them amongst the offspring even being born.
So, after watching this video and the discussion that we have done so far, the main key takeaways about the lethal alleles are an inheritance pattern in which an allele is only lethal in the homozygous form and in which the heterozygotes may be normal or have some altered or non-lethal phenotype is called as recessive lethal. The dominant lethal inheritance pattern is the one in which an allele is lethal both in the homozygous as well as in the heterozygous condition, and this allele can only be transmitted if the lethality phenotype occurs after the reproductive age. Dominant lethal alleles are very rare because the alleles only last one generation, which means that therefore they are not transmitted. And in the last, the case where dominant lethal alleles might not be expressed until adulthood, the alleles may be unknowingly passed on, resulting in a delayed death in both generations. Now let's look at the phenomenon of expressivity and penetrance. First of all, let me tell you what is penetrance. Penetrance refers to the probability of a gene or a trait being expressed. In some cases, despite the presence of a dominant allele, a phenotype may not be present. One example of this is polydactyly in humans, which means they have an extra finger or an extra toe, sometimes more than 6 or 7. A dominant allele produces polydactyly in humans, but not in all humans with the alleles display the extra digit. Complete penetrance means that the gene or the genes for a trait are expressed in all the population who have that gene, while incomplete or reduced penetrance means that the genetic trait is expressed only one part of the population. Penetrance of the expression may also change in different age groups of the population. Reduced penetrance probably results from a combination of genetic, environmental, and lifestyle factors, many of which are completely unknown. This phenomena can make it challenging for the geneticist professionals to interpret a personal family medical history and predict the risk of passing a genetic condition to the future generations. Expressivity, on the other hand, refers to a variation in phenotypic expression when an allele is penetrant. Back to the polydactyly example, an extra digit may occur on one or more appendages, which means on one hand or on the other hand. The digit may be of full size or it can also be a little stub. Hence, this allele has reduced penetrance as well as variable expression or expressivity. Variable expressivity refers to the range of signs and symptoms that can occur in different people with the same genetic condition. As with reduced penetrance, variable expressivity is probably caused by a combination of genetic, environmental, and lifestyle factors, most of which have not been identified yet. If a genetic condition has highly variable signs and symptoms, it may be challenging to diagnose. Let's look at an example of penetrance and expressivity in Drosophila.
For the gene Antennapedia, whose mutations can cause flies to develop legs coming from their heads instead of antennae, if this followed a purely Mendelian pattern of inheritance, then when a fly which is homozygous for mutant Antennapedia genes is mated to wild type flies which are homozygous, then the offspring should either be wild type in phenotype or Antennapedia, depending on which allele is dominant. Instead, we notice two phenomena. First, some of the flies of the F1 generation possess the Antennapedia trait, and others do not and are wild type. So thus, this mutation is not fully penetrant, because flies with the same genotype may not express the same phenotype. Secondly, the Antennapedia trait can be expressed to varying degrees. The size of the legs and the degree of malformation of the antennae of those mutant flies can vary. One fly can be different from the next. And so therefore, while they all share the same mutation, the overall phenotype can vary. So therefore, the expressivity can vary. And so Antennapedia varies in both penetrance, in that not all flies which possess a mutant allele will express the Antennapedia trait, and also in expressivity, because different flies with the same genotype may express a mutation to varying degrees.
So, the key takeaways or main points related to penetrance and expressivity are: penetrance is used to describe whether or not there is a clinical expression of the genotype in an individual, while expressivity is the term that describes the differences observed in the clinical phenotype between two individuals with the same genotype. Now let's look at gene interactions. Gene interactions occur when two or more allelic or non-allelic genes of the same genotype influence the outcome of a particular phenotypic character. To understand the molecular basis of this complex biological phenomena, there is a need of genetic interaction mapping where the effect of one gene are modified by one or several other genes. Genetic interaction is a set of functional association between the genes. One such relationship is epistasis, on which we are actually going to focus in this lecture. In case of epistasis, the interaction is between non-allelic genes, where the effect of the one gene is actually masked by the another gene, which results in the suppression of the effect of one, or the both of these are going to combine to produce a new trait or a new character. Gene interactions are mainly of two types. We have intragenic gene interaction, and then we have intergenic gene interaction. In case of intragenic gene interaction, two alleles of a gene which are present on the same gene locus on two homologous chromosomes, they react to produce a modified phenotype. Phenomena that include intragenic gene interactions include incomplete dominance, codominance, multiple alleles, and lethal alleles. Then we have intergenic interactions. In intergenic interaction, modifications of the effects of a genes are under the influence of a non-allelic gene which is present at some other chromosome, not on the homologous chromosome, or maybe on another locus of the homologous chromosome. Now, this is called as intergenic interaction between because the genes which are actually affecting each other are present at different locus. As we have already discussed about the gene interactions, which come under the heading of intragenic gene interaction, which were codominance, incomplete dominance, multiple alleles, and lethal alleles. We are actually going to focus in this lecture on the intragenic gene interactions, and our main focus is going to be on epistasis. But the phenomena that come under the intergenic gene interaction include complementary genes, supplementary genes, epistasis, which has many other types, and I'll be explaining these in the later on slides as well, and duplicate genes and pleiotropic genes. So first of all, the complementary genes. These genes are the genes which are present on different genetic loci, but they interact with each other to express a single character in combination. They both together produce a particular phenotypic trait in an individual. In case of the supplementary genes, these are two non-allelic genes in which the first gene can produce its effect whether the second gene is present or not. But the second, which is called the supplementary gene, actually produces its effect only in the presence of the first gene. Then we have the main thing, which is epistasis. Epistasis is the interaction between the genes present at two different locations or loci in which one gene suppresses or masks the expression of the other gene. So since there is a gene that is being expressed and the other one that is being suppressed, so we have two types of genes which come under this phenomena of epistasis. We have epistatic genes and then we have hypostatic genes. Epistatic genes are the genes that determine whether or not a trait will be expressed. So these are the main controllers or the boss. Then we have hypostatic genes. Hypostatic gene is the one whose phenotype is altered or changed by the expression of an allele at a separate locus, which was the epistatic gene in an epistatic event. Then we have duplicate genes. Duplicate genes are two identical genes showing the same phenotypic action, but they are localized in different regions of a chromosome or on different chromosome, and it is known as a duplicate gene. Then we have pleiotropic genes. When a gene affects many aspects of the phenotype or controls several phenotypes, it is said to be pleiotropic gene, and the phenomena is called as pleiotropy. Now let's look at epistasis in detail. Epistasis. As I told you, epistasis is the interaction between the genes present at two separate loci or locations in which one gene suppresses or masks the expression of another gene. The gene controlling the expression of the other gene is called epistatic gene, while the gene that is being suppressed by the epistatic gene is called the hypostatic gene. Now, in the phenomena of epistasis, we have two types of epistasis. Actually, we have three types, but you need to focus on two of them. One is called as dominant epistasis and the other one is called as recessive epistasis. In case of dominant epistasis, when a dominant allele at one locus can mask the expression of both alleles, which is dominant and recessive, of another gene at another locus, it is known as dominant epistasis. In simple words, you can say that one copy of epistatic allele is sufficient to mask up the effect of the hypostatic genes. And I'm going to give you some example of in detail as well. But a very simple example is that of different colors of the summer squash. That's an example of dominant epistasis. Well, in case of recessive epistasis, it is a form of epistasis in which a pair of recessive alleles of one gene can cause a mask effect on the expression of the alleles of another gene at another locus. Or in other words, we can say that two copies of epistatic allele is necessary to mask the effect of the hypostatic allele or gene. A very good example is that of coat color in Labrador dogs. Here is the example that I'm going to explain in the phenomena of dominant epistasis, and I gave you the example of different colors which are found in summer squash. We have two genes here, gene 1 and gene 2. Here gene 1 is the epistatic gene, whereas gene 2 is the hypostatic gene. So gene 1 is the controller and gene 2 is the slave. So in gene 1, you see for pigment deposition, we have the dominant allele, which is being represented here by capital letter or uppercase letter W. And the recessive one is being represented by a small w. Here, capital W, it does not allow color pigment deposition, whereas if the recessive allele is present, it allows color pigmentation deposition. Now, the hypostatic gene here is responsible for the color of the squash. If it is uppercase Y or capital Y, the color is going to be yellow. And if it is small y, the color is going to be green. So, If we have a combination in which capital W is present with the small w and small y or capital Y, the controller gene W is not going to allow color pigmentation, whether small w is present with it or capital Y or small y is present with that. So in any combination, if capital W is present, it's not going to allow color pigmentation. Only one dominant allele is responsible for controlling the effect of all other alleles, and that's an example of dominant epistasis. So let's look at an example of recessive epistasis, and a very good example is Labrador dog coat color, and you see that they come in a variety of colors, and the color coat may range from pitch dark black to different shades of brown and to golden yellow. Okay, so there are two genes which are involved in the coat color. We have gene 1, which is responsible for the coat color. If it is capital B allele, it's going to be black. If it is small b, then the coat color is going to be brown. Then we have another gene here, which is playing a very important role, an epistatic role in deposition of the pigment in the hair shaft of the dog. So we have two copies of that gene, capital A, the dominant, which allows dark color pigment deposition in the hair shaft, and small a, which is the recessive one, and it does not allow that. So, as I told you that in case of recessive epistasis, we need two copies of the epistatic alleles in which is necessary to mask up the effect of the hypostatic gene. So we have gene number 2, which is the epistatic gene, which is actually controlling the coat color, which is the gene 1. So if this gene, epistatic gene, is present in a homozygous condition, we have just two copies of A, whether it's capital A or small a. It's not like going to allow pigment deposition in the hair shaft of the hair of the Labrador, as a result of which we will have a yellow Labrador dog. So how the cross work? Let's look at that. So here you see that we are crossing heterozygote for the B gene and A gene. We have the dominant and recessive copies of both, and we cross them. We get a ratio of 9:3 is to 1 of black to yellow to brown and to yellow. And you see that in the yellow one we have AA, which is the epistatic gene. It is, although it is the recessive allele, but it is present in a homozygous condition. It's not letting the color to be deposited, and even if capital B is present there, the dog is yellow. Similarly, in the last row, you see that you have two copies of small a and two copies of small b. Both of them are homozygous for the individual alleles, and you can see here that once capital A, capital small a, and small a is present in a homozygous condition, two copies of that are present. It's not letting the color to be deposited, the pigment to be deposited, and the dog has a yellow hair color. So after looking at this cross, let's watch a video which is explaining the phenomena of epistasis with the same examples to understand it in a very nice way with the same examples.
Epistasis may seem like a complex concept, but it can be reduced to a few simple moving parts. This doggy represents one gene called B. Epistasis occurs when another gene called A affects its expression. Think of A as holding B on a leash. We will see that the leash can come in different types. Why is there epistasis? Well, epistasis can emerge anytime in most single gene traits. Take, for example, formation of hair on the leaf surface of the plant Arabidopsis. A gene called H might have a recessive allele, little h, that confers hairless appearance. As predicted by Mendel, when heterozygous big H little h is selfed, we get a good 3:1 ratio of hairy to hairless. Is this a nice single gene trait? Not really. We know from multiple experimental evidence that the H gene works together with many other genes, as represented by this network, where every ball is a gene and every line represents an interaction between two genes. Therefore, while the action of a gene may appear isolated, no gene is an island. Instead, it works together with many other genes. When variation as these other genes occurs, epistasis can result. Let's see how it works. I will use 3 examples where gene A is epistatic on gene B to illustrate 3 cases: recessive, dominant, and double recessive epistasis. The real gene names may be different in reality, but I will stick to A and B for comparison. In all cases, I will deal with the phenotypes in F2 individuals formed by selfing a double heterozygote with big A little a, big B little b genotype. The following assumptions apply. Big A is dominant to little a, big B is dominant to little b. The A and B loci are unlinked. We expect 4 phenotypic classes: one that has at least one big A and one big B allele, another with at least one big A and little b, a third with little a, and at least one big B, and a fourth with little a, little b, the double recessive homozygote. The underlined mark refers to either of the 2 possible alleles. In all cases, B can act as a regular Mendelian gene affecting a character for which there is a dominant and a recessive trait. Let's start with recessive epistasis. Big B confers black coat in Labrador retrievers, little b, brown coat. The big B allele encodes a functional enzyme that converts a brown pigment into a black pigment. The little b allele encodes an inactive form that cannot produce the black pigment. This would be simple, but there is a catch. A second gene, big A, is epistatic on B. It encodes a protein necessary for deposition of the pigment. If the big A protein is not present, that is, if little a is homozygous, the coat is yellow. So let's consider the 4 classes. If the big A and big B alleles are present, black pigment is formed. If big A is present, but not big B, brown pigment is formed. If big A is absent, yellow coat results. The expected ratio is 9 black to 3 brown to 4 yellow. Let's look at these phenotypes in a 4x4 Punnett square where the gamete types are marked on the sides. Yellow appears in 4 square where the B gene action does not count because of the absence of big A. The other squares are free to undergo whatever color change the B genotype dictates, such as brown and black. If you step back and look at the whole layout, you see that 4 squares out of 16 display the yellow epistatic condition. Therefore, we call this recessive epistasis. Let's now consider dominant epistasis in squash. The setup is very similar to that of the black lab dog. The B gene encodes a protein that converts green pigment into yellow. The accumulation of pigment, however, is controlled by an epistatic gene called A. However, instead of promoting pigment accumulation, A is a repressor that blocks it. Pigment can only be formed in the absence of the big A allele, such as when the little a allele is homozygous. Let's look at the 4 phenotypic classes. The presence of big A results in white squashes. In the absence of the big A repressor, big B results in yellow squashes, and little b in green squashes. The F2 ratio is 12 white, 3 yellow, and 1 green. In the Punnett square, we mark green the double homozygote recessive, yellow the 3 squares that have big B but lack big A, and the rest are white. Looking at the layout, the epistatic big A allele affects 3/4 of the 16 squares in a typical dominant ratio. Last, let's consider the case of double recessive epistasis in the snail. Here, the B gene converts colorless precursor 2 into a dark pigment, resulting in a dark shell. The A gene is epistatic because it is needed for the conversion of upstream colorless precursor 1 into precursor 2. Consider the pathway. It yields dark pigment only if both the A and the B genes have the dominant, that is, active allele. The F2 progenies display two phenotypes, dark and albino in a 9:7 ratio. Looking at this in the Punnett square, we mark as dark the squares where both big A and big B are present. The rest are albino. The albino F2s are laid out in an unusual pattern. This, however, makes sense. The 4 square marked in orange represent the 4 out of 16. If we now exclude these, the remaining 3 albino squares are 3 out of 12. Therefore, a 3-to-1 ratio is displayed twice, as one may expect in a double recessive situation. In summary, an organism is a complex structure whose traits depend on chain of events, each controlled by a different gene. Epistasis results when variation in genes required for upstream events affects the events downstream.
The key takeaways from from this phenomena of epistasis that I have discussed and the videos that you have watched, the main points are that in many cases several genes may contribute to a particular phenotype. When the action of one gene masks the effect of another, the phenomena is known as epistasis, and the gene is said to be epistatic to the second one. Epistasis can occur when a recessive genotype masks the action of another gene or when a dominant allele masks the effect of another gene. Epistasis can be reciprocal, either gene when present in the dominant or in the recessive form expresses the same phenotype. Any single characteristic that results in a phenotypic ratio that totals to 16, such as 12 ratio 3 ratio 1 or 9 ratio 3 ratio 4 or other, is typical of a two-gene interaction.